everyone, my name is Kim. I'm Assistant Professor of Choi University School of Medicine and first author of this article. It is a great privilege for us to have an interview with other section in ICUrology and appreciated to be awarded in 2019 annual meeting of KUA as best paper on basic research. Yong and Yoon, the second author, is Assistant Professor of Hanyang University College of Medicine. Professor Hong-Sam Moon, the corresponding author, is the chairman of Urology Department of Hanyang University. This article is entitled Carbon Monoxide Releasing Molecule 3, Amelioration of Renal Ischemia-reperfusion Injury in a Rat Model. For background information, previously ischemia-reperfusion injury was studied in cardiac, cerebral, hepatic disease. In urology, ischemia-reperfusion injury could occur in kidney transplantation or partial nephrectomy. Recently, partial nephrectomy usually performed by laparoscopic or robot-assisted surgery. Given the detrimental impact on kidney function by warm ischemia time, it is important to manage ischemia-reperfusion injury in a kidney cancer as well. For the mechanism of this issue, ischemia-reperfusion injury occurs when target organ has reperfusion after ischemic time. Membrane injury causes calcium increase at mitochondria, which decreases ATP production and makes cell injury. Therefore, the purpose of this study is to investigate the in vivo effects of carbon monoxide releasing molecule 3 on ischemia-reperfusion injury using rat models. Materials and methods. Rats were divided into 3 groups. Animals in the sham group underwent right nephrectomy and the abdominal cavity was open and closed. Animals in the ischemia-reperfusion injury group underwent right nephrectomy and occluded the left renal artery and vein. And also for animals in the CORM-3 group were injected with CORM-3 in the tail vein 1 hour preoperatively and performed ischemia-reperfusion injury as well. Kidney tissues and blood sample of rats were evaluated. Serum creatinine level, renal injury markers, tunnel assay, pathologic results, cDNA microarray were investigated. Here is the results. The ischemic time on 75 minutes, CORM-3 group revealed significantly lower serum creatinine level compared to the ischemic group. The renal injury markers, such as kidney injury molecule 1 and cystatin C level, revealed significantly lower compared to the ischemia control group. Here we can see pathological results. H&E staining revealed normal tubal appearance of the kidney tissue in the sham group. However, the ischemia-reperfusion injury group exhibits severe tubular dilation and necrosis of the tubular epithelium. Conversely, the CORM-3 group showed only partial tubular thinning and no tubular necrosis. The tunnel assay was performed to determine apoptotic activity in the kidney. The sham group exhibited few apoptotic cells. However, the level of apoptosis increased markedly in the ischemia control group, and the CORM-3 administration reduced the number of apoptotic cells. We studied the pathways involving genes with aberrant expression in the ischemia control group and identified 5 pathways, including the metabolic pathway and PI3K AKT signaling pathways in the CORM-3 group. The expression of these genes was poor or not detected. In the ischemia control group, 3,746 genes showed altered expression, while the expression of only 960 genes was altered in the CORM-3 group. A total of 15 genes were found to be involved in the peroxisome proliferator activated receptor signaling pathway, including MCAD, LXR alpha genes, and the difference in the expression of these genes was statistically significant. In order to analyze changes in the expression of genes involved in the PPAR signaling pathway in the sham, ischemia-reperfusion injury group, and CORM-3 groups, gene-specific primers were designed and RT-PCR was performed. The expression of PPAR pathway genes was higher in the CORM-3 group. The CORM-3 inhibit ischemia-reperfusion injury by upregulating genes associated with the PPAR signaling pathway. PPAR signaling pathway has a protective effect on ischemia-reperfusion injury, and it is also involved in lipid metabolism, gluconeogenesis, and also for cell survival. CORM-3 pretreatment can reduce cell deaths and inflammatory responses and increase cell growth. The result of our analysis reveals that CORM-3 pretreatment can preserve renal function. And also CORM-3 reduces renal ischemia-reperfusion injury through the upregulation of genes involved in the PPAR signaling pathway. So in conclusion, CORM-3 inhibits the progression of acute renal injury by reducing inflammation and apoptosis. The PPAR pathway plays an important role in the mechanism of CORM-3. So we expect that pretreatment with CORM-3 in partial nephrectomy or kidney transplantation could minimize renal ischemia-reperfusion injury. So thank you for reading our article.